Alright guys, so this lesson is going to be moving forward in our new unit. So up to this point we have covered enzymes and how they regulate our metabolism. And so we're going to have two more lessons in this unit, and both of the lessons are going to be about two major metabolic pathways. And so in our cells we know that we have all these chemical reactions. We have some that are building molecules, we have some that are breaking molecules. Those that are building are storing energy, and those that are breaking are releasing energy. And so this particular lesson is about a pathway that would be considered catabolic. And what that means is we're going to be breaking down materials in order to release energy that our cells can use. And that process is called cellular respiration. Hopefully that is familiar to you, because when we talked about cell parts, hopefully we know that the mitochondria, the reason we call it the powerhouse, is because the mitochondria makes energy for our cells in the process of cellular respiration. And so all living things need energy. It doesn't matter what kind it is. It doesn't matter whether it's a simple bacteria or a complex animal or plant. All living things have to have energy, and they get that energy when they break down food molecules. Okay, so remember that all organisms have to consume or make food molecules. That's where we get into our autotrophs and our heterotrophs. So it doesn't matter where they get it from, but once they get that food, they're going to have to take it and they're going to have to break it down. Because when we break down these chemical compounds, we release energy when we break those chemical bonds. And so the type of energy that we're dealing with here is something called chemical energy, and that just is energy that's stored in bonds. So anytime we break those bonds, energy can be released. Now technically we can use any of these molecules to produce energy for our cells. But back when we covered macromolecules, hopefully you know that carbs are our main energy source. We can also use fats. They can store quite a bit of energy. And then proteins, I mean, technically you can break any bond to get energy, but we really don't want to be breaking down proteins in our body. We don't want to break down muscles, or we don't want to break down enzymes in order to get energy. So even though these could be used, we don't necessarily want to use these as a basic fuel for our body. So it's those simple carbohydrates like glucose that serve as our main energy source. And this is what we're going to see in the reaction of cellular respiration. Now, in the event that glucose and simple carbs were not available, we could tap into our fats and proteins and use those to produce energy in our body too. So we have to take that food and we have to take those nutrients, like I said, especially that glucose molecule, and we have to turn it into a form that our cells can use. So we said that glucose is energy, and the reason is that is going to be the chemical energy form that we are going to break in order to produce another chemical energy form called ATP. So when we talk about energy for our cells, what we're talking about is this little molecule. It's actually kind of a big molecule, but it's called adenosine triphosphate, and that's where we get ATP, okay? So essentially what we're doing in cellular respiration is we're going to take the glucose and we're going to produce ATP, which is what makes the mitochondria the powerhouse. So exactly what is this ATP molecule? Well, it's actually a nucleotide, and this also goes back to our macromolecules. Nucleotides were the building blocks for things like DNA or RNA, but this is actually a special nucleotide in that it's not just like the ones that would go in DNA or RNA. It's actually very similar to an RNA nucleotide. It has the ribose sugar. It has the base adenine. But what makes it different from a DNA or RNA nucleotide is that instead of just having one phosphate group, it has 3 phosphates. And so this right here is what makes this molecule so special. Okay, it's this region. So here's my ribose sugar, just like an RNA nucleotide. Here's my adenine nitrogen base. This is just like the adenine when we do A, T, C, and G. This is your A. But notice that I have 3 phosphate groups over here. Now the thing is this is where we get our energy from. Now we're not going to go into it in too great a detail, but what I need you to understand is that these phosphates are unstable. They all carry a negative charge, and negative does not like negative. And so when they're bonded, they're pretty unstable bonds, and unstable equals high energy, especially this third one, okay? So in this third phosphate bond here, there's a lot of unstable, high energy, ready to explode. And so all that has to happen to release that energy is that third phosphate has to be broken off. And it's actually broken off by that process that we talked about called hydrolysis. So the way ATP gives energy to our cells is it breaks off that third phosphate, and that releases energy that the cells can then use to do different activities. And so one thing I want you to understand too is the ATP cycle. So because this ATP is unstable, we're not really storing it, okay? This is not a storage molecule. We pretty much make ATP on demand as we need it. If we are doing more activities that require more energy, then we are going to be making more ATP in that moment. If we're not doing as much, then we're going to be making less ATP. So it's kind of a made on demand kind of molecule. And so the way it works is ATP has 3 phosphates. When it loses a phosphate, it becomes a molecule known as ADP. So this was a triphosphate. This is diphosphate. So it becomes ADP, which only has the 2 phosphates because one of them was lost. It was broken off. It was transferred to something else. And so the process of going from ATP to ADP releases energy for the cell to use. Now, in order to make ATP, what we do is we do the opposite. We're going to have to add a phosphate. So we take a phosphate group and we add it to the ADP. Now, hopefully we know that in order to form a bond, in order to make a bond, it's going to require some energy. So we have to have a little energy to make energy, if that makes sense. And so the process of cellular respiration is going to be how do we get the energy to add phosphates onto ADP. And so what we have is this constant cycle, okay? So the ATP is made when a phosphate is added. It's unstable. It's quickly broken. We lose the phosphate. It becomes ADP. That stores the energy, okay? Or that releases the energy. We add a phosphate back on. That stores the energy. It breaks. It releases the energy. And so what we have is this ongoing cycle. So when these ATPs are made, once again, they're not around for very long. It's going to be quick, okay? They're going to be made, and then they're going to be broken. And then they're going to be made again, and then they're going to be broken. So anytime it's made, it has 3 phosphates. The third phosphate breaks off. It becomes ADP. Then you add the phosphate back. It becomes ATP, and so on, and so on, and so on. And so here's some of those vocabulary words that we should have learned from our last lesson. So energy from catabolism is going to be used to make the ATP, and then the energy that's released is going to be used for anabolism, or those endergonic reactions. Now, why is ATP so important? Well, it pretty much powers our cells. It can be used for things like active transport. Anytime we're pumping molecules where they don't want to go, it can be used to synthesize other molecules like protein synthesis. We use a tremendous amount of ATP in muscle contraction. We recycle like 1 million ATPs per second or something like that. I mean, it's a ridiculous amount. And so we need these ATP molecules for all different types of activities in our cells. So because we can't store it, what we do, because that's unstable, remember, and so what happens is what we do is we store those other molecules, like we can store the carbs in the form of glycogen, or we can just take those glucose molecules from our blood, or we can break down fat. So we have these storage molecules that we use when we need them on demand to make those ATP molecules. And so the process, the main process that we use in our cells to make energy is going to be cellular respiration. This is going to be the most efficient. This is also known as aerobic respiration because it requires oxygen. And so this is the chemical reaction for cellular respiration. This is something that you just need to be familiar with, okay? Because we're going to see something very similar to it in our next lesson when we cover photosynthesis. So if you don't know what these molecules are, you do have the word form right down here. You should know it both ways. But cellular respiration is a process where we take glucose and we break it down in the presence of oxygen. And in that process, we produce some carbon dioxide, we produce some water, and we produce, most importantly, energy in the form of ATP. Now, some of that energy is going to be lost as heat. It's not going to be a perfect process. This is one of those random forms of energy, but we will generate some heat as well, okay? But this is what it's all about, is making this molecule right here, ATP. So glucose is broken in the presence of oxygen to produce carbon dioxide, water, and energy. Now, it looks like it's just one chemical reaction, but that's a little bit deceiving because, in fact, it is a series of many smaller steps. If we just blew that glucose molecule up in one big step, most of that energy would be wasted in the form of heat and light. It would kind of be almost like a combustion reaction, and we would lose a tremendous amount of the energy, and it would not be available to the cells. In fact, it would harm the cells. And so what we do is we break it down in a series of processes, okay? You can see that it's not just one big reaction, it's actually several reactions put together. And by the way, this right here would be a great summary slide. You might want to make a note that on this particular slide there's a great summary. This is what we're going to be going through over the next several slides. Now, I also want to give you a little heads up. Some of this is in your notes, some of it's not. But cellular respiration is not an easy concept to learn. And the reason why is it's very chemical, okay? What you have to understand is that it is a series of chemical reactions. And the reason that these chemical reactions are happening is because these molecules just happen to be in the same place at the same time. And we know that atoms react based on their properties, okay, based on their valence electrons. So all of this is happening just because of the chemicals that are in our cells. We can't really see it. It's very abstract. And so I know that it can be difficult for students to learn, okay? I know that it is not an easy concept. So I kind of broke it down for you. What do you really need to know? As we go through the rest of this lesson, we're going to have three main processes. All right, they were listed in that little summary just a minute ago. So we're going to talk about three main processes that make up that cellular respiration reaction. You need to know where they happen in the cell in terms of like organelle or cytoplasm. And you need to know what's going into each stage and what's coming out of each stage, okay? So even though I know it can be difficult to learn, I want you to focus on these important things. What are the processes? Where do they happen? And what's going in and out of each process, okay? So as we go through this, there's one other thing that I need to go over in order for this to make sense to you, okay? We do need to learn a little something about electron carriers, because as we go through the three processes, one of the things that we will be keeping track of are electron carriers. And so once again, it's very chemical, but I want to remind you that electrons are energy, okay? They are the negatively charged particles. They're moving around out in that cloud, and electrons have energy. So when we move electrons from one atom to another, it's carrying energy with it, okay? So just when they move into different orbitals, they're changing energy levels, all right? That's what those orbitals are. So when they get transferred to something else, they're carrying some of that energy with them because electrons are energy. So we're going to talk about two electron carriers. And here's what you need to understand about electron carriers. They are carrying electrons or energy to be used later, okay? The reason we're keeping up with these as we go through the processes is they're carrying energy in the form of electrons to be used later. Now, the electrons don't go by themselves, okay? So when we talk about these electron carriers, we have two main examples. We have NAD and FAD. They are both coenzymes. Those are some of those helpers to enzyme activities. And when they shuttle electrons, when they actually accept them and then carry them, they don't just carry the electrons by themselves. The electrons come along with a hydrogen atom. So a hydrogen atom is a proton and an electron, and then it'll bring electrons with it, okay? So when we are carrying electrons with these molecules, I want you to notice what they become. They become NADH and FADH2, okay? So these right here are what they are when they don't have the electrons that they're carrying. This is what they are when they do have the electrons. So they're carrying the electrons along with a hydrogen there, okay? So the reason we're keeping up with those is because they're carrying energy to be used later. And we'll talk about when that later is. So here's just some pictures to kind of help you understand NAD and FAD, okay? So all they're doing is just carrying electrons and a hydrogen. When they're carrying electrons, they now have the H, okay? They kind of lose that charge. So you can see that I have a proton and a couple electrons here. Here I have a couple of protons and a couple electrons here. And so in this form, they're just carrying some energy for us to be used later. All right, so once again, there's three main things that you need to know. Okay, this right here is a summary of several of them. Number one, you need to know the three main steps. And the three main steps are glycolysis, the Krebs cycle, and the electron transport chain. The second thing you needed to know was where do they happen. Okay, glycolysis happens in the cytoplasm. The Krebs and the electron transport chain are in the mitochondria. Okay. Now, in terms of the input and the output, we're going to go into each of these in more detail. Once again, here's a great summary of the whole process. So I would encourage you to pay attention to the pictures that are in your notes. You can see that glucose is going into glycolysis. Okay, we have some things going in, some things coming out. All right, we have the Krebs cycle, some things going in, some things coming out, and so forth. So we're going to start with the first process, which is glycolysis. And let me tell you, if you think this is hard, please know that it could be way worse, okay? So I want you to notice that glycolysis is actually 10 enzyme catalyzed reactions. All right, so just the first stage of cellular respiration is a series of 10 reactions. Each one is catalyzed by an enzyme. And if you were in biology for majors right now, one of the things you have to do is know the products of each and the enzymes that catalyze it, okay? What you need to know here is the big picture, okay? Glycolysis is a series of reactions. Now, it is considered an ancient pathway. Glycolysis is believed to be the first energy-producing pathway like this. And so it existed before we had eukaryotic cells. It existed before we had mitochondria. It existed before we had an oxygen-rich environment. And so this particular pathway, the very beginning stage, this particular one does not require oxygen, and this one is in the cytoplasm. Okay, pretty much every organism on the planet is going to go through the process of glycolysis. They don't have to have a mitochondria. They don't have to have an oxygen-rich environment. So glycolysis is the starting point for all pathways, both aerobic and anaerobic. And so as we go through the steps of glycolysis, once again, there's technically 10 steps. What you need to really understand is that we start with the glucose. Okay, so here's my starting point. Now, I want you to notice that glucose is a 6-carbon sugar. Remember, it's C6H12O6. So here I have my 6-carbon glucose. And in this series of 10 steps, the glucose is broken down into 2 pyruvates. And so here we see 1, 2, 3 carbons, 1, 2, 3 carbons. So essentially what we're doing is we are splitting a 6-carbon sugar into 2 3-carbon compounds. Okay, so glucose becomes pyruvates. Now, in that process, we use 2 ATP. This is what we call the energy investment phase, but we produce 4. And so for glycolysis, we have to take into account what we call the net gain. It's kind of like your paycheck. You have a certain amount of money that you made, but you do have to subtract things like taxes, okay? And so what you bring home is not actually what you made. This is kind of what's going on here. So even though we made 4 ATP, we're only gaining 2 total because we used 2 in the process. Now, 2 ATP at a time, this is not very efficient. All right, imagine that I gave you 2 pennies and said, hey, go buy yourself something nice, okay? You can't really do much with 2 pennies. It's going to take a lot more energy for our cells to be efficient, okay? So this is just the starting point. So once again, what do you need to know? The process is glycolysis. This is happening in the cytoplasm. What's going in? Glucose. What's going out? Okay, so we got 2 pyruvates, those 3-carbon compounds. We gained 2 ATP, that's our net gain. We also got a couple of electron carriers, okay? And these electron carriers, remember, are carrying electrons to be used later. So that's why we're going to be keeping up with them. All right, so now we're ready to kind of go to the next pathway. And this is the branching point. Once we go through glycolysis in the cytoplasm, at this point, we're either going to go the aerobic pathway, which uses oxygen, or we're going to go the anaerobic pathway, which does not use oxygen. Now, we're going to be starting with the aerobic pathway, and then we're going to come back to the anaerobic pathway at the end of the notes. So for the aerobic pathway, this is what's going to happen if oxygen is available. And at this point, we're going to move into the mitochondria. So this is why we call the mitochondria the powerhouse, because these main ATP producing processes are happening in the mitochondria. So those pyruvates that we got from glycolysis are going to enter into the mitochondria, and we're going to continue their breakdown, and in that process we're going to make some more ATP. Okay, so we are now going the anaerobic route, and when we talk about the mitochondria, we're going to be mentioning some specific locations. So this is just a little bit more information, maybe a reminder of the parts of the mitochondria. So it does have a double membrane, so we have an outer membrane on the outside and an inner membrane on the inside. Now, that inner membrane is highly folded, okay, and we call those folds cristae, okay, or cristae, however you want to say it. And so we have the outer membrane and the inner membrane. Now, between the two is something called intermembrane space, all right? And so that's just going to be the space in between. And then we have the liquidy stuff on the inside, and that's called the matrix, okay? And in the matrix, you have the fluids, you have some DNA. Remember that your mitochondria are kind of special organelles. They have their own DNA, they have their own ribosomes, they're going to have all these enzymes that are needed. And so we have the two membranes, the inner one's highly folded, and then we have the matrix, which is the liquidy stuff on the inside. So before we can actually go into the next step, which is the Krebs cycle, there's a little bit of an intermediate step here. Okay, so those pyruvates that we made, remember they were 3 carbons. Okay, here's 2 pictures. They're both showing the same thing. So here's my 3-carbon pyruvate or pyruvic acid. What we have to do is we have to turn it into another molecule called acetyl-CoA. This is acetyl coenzyme A is what that stands for. And so in order to do that, what happens is we lose a couple of carbons. Okay, we're going to go from a 3-carbon pyruvate to a 2-carbon acetyl-CoA. All right. So all you have to really know here is that it's the acetyl-CoA that's going to be going into the Krebs cycle. Now, in this process of going from pyruvate to acetyl-CoA, I want you to notice that I lose some carbons. We had 2 pyruvates. Each one is going to lose a carbon. And so this is where a couple of our carbon dioxides come out. Remember, that's on the product side of our reaction. We also get a couple of more electron carriers. Those are going to be used later. And what we really needed for this were the 2 acetyl-CoA. Okay, that's the molecule that's going to enter into the Krebs cycle, which is technically our second main stage of cellular respiration. Now, this is also known as the citric acid cycle because citric acid, this little gray molecule here, this is the first product in the Krebs cycle. It's called citric acid. This is the same acid that you find in citrus fruits, like oranges and lemons, okay? So you can see that it's a 6-carbon compound here. Now, once again, what looks like one process is actually 8 steps. We have enzyme catalyzed reactions all the way around. You don't have to know the individual products. What you need to understand is that in the process of the Krebs cycle, we're going to completely break down that glucose molecule. By the time we are done with the Krebs cycle, all of our carbons will be lost in the form of CO2. So we already released 2 in that little intermediate step, and here's where we're going to release the remaining 4. Okay, so if you go back to that original equation, you had 6 CO2. This is where 4 of them came from. The other 2 came from the previous step. Now, what you need to know are the processes. So the process name is called the Krebs cycle. Where does it happen? We are now in the mitochondria, specifically the matrix, that liquidy portion. Now, big idea here, we made 2 ATP, okay? We also made some more electron carriers to be used later. So there's your input and your output. We have the Krebs cycle. It's taking place in the mitochondrial matrix. Now, what went in are acetyl-CoAs. Okay, that was from that intermediate step. Remember that we had 2 of them. We had 2 pyruvates, and we have 2 acetyl-CoAs. Because there are 2, the Krebs cycle will have to happen twice, okay, in total. So 2 acetyl-CoAs went in. I produced 4 CO2. And guys, those are just going to be exhaled. This is right here is why we exhale CO2. So those are just going to be exhaled from our body. I get a bunch more electron carriers. Okay, remember that these are carrying energy to be used later. And then I get 2 more ATP. So each turn of the Krebs cycle gave me 1 for a total of 2. Now, once again, still not a lot of energy. If I gave you 4 pennies and told you to go buy something nice, you still really can't buy a whole lot. And so now we're going to enter into the last and final stage, and it's known as the electron transport chain. The electron transport chain is a chain of proteins, okay? So we are now in the mitochondrial cristae, or that inner membrane. So those are those little membrane folds that we looked at. And so these are just proteins that are embedded in that membrane, okay? Now, this is where we're going to use that to be used later. Those electrons that have been carried by our electron carriers, they're going to be important here. And they're going to help us produce a lot more ATP. Okay, at this point we made 2 in glycolysis and 2 in the Krebs. Notice that in this process I'm going to get about 32 to 34. So that is a lot more energy than the other processes. Okay, now this right here is also why we need oxygen. Oxygen drives the whole process of the electron transport chain. So this actually goes back to a lesson that we did in chemistry. When we were covering polar and nonpolar, remember that some atoms like electrons more than others? Well, oxygen is one of those that really likes electrons. It has a very high electronegativity. And in the case of cellular respiration, oxygen is what we call the final electron acceptor. Oxygen is the driving force behind this whole process. Without oxygen, this process stops. And without oxygen, you die, okay? This is how important oxygen is. This right here alone is why we have to breathe in oxygen to survive. Because if we don't have oxygen, we can't make very much ATP. And if we don't have ATP, our cells can't function properly, okay? So at this point, we made 2 ATP in glycolysis. We made 2 ATP in the Krebs cycle. And now with the help of oxygen, we're going to make a whole lot more, okay? So this is where our electron carriers are going to come into play. They're going to drop off their electrons and the hydrogens that they've been carrying. I'm just going to be honest with you guys. This is a very, very, very technical process. There's a lot of important concepts here. I have tried to simplify it to the best of my ability so that it makes sense to you, okay? I can't promise that it's still going to make sense. We're going to have to do lots of practice with it. I'm going to encourage you to watch videos on it. But this is just one of those things that you may have to spend some time with to understand it, okay? So we're going to start with those electrons. Remember that these electrons were carried by the electron carriers. And like I said, electrons are energy. The reason they do this is because of oxygen. Because oxygen is present in that mitochondria, they are going to get pulled through the electron transport chain, okay? So I've tried to break it down into as simple a terms as I possibly can. So the electrons get dropped off and they get pulled through those proteins of the ETC. Like literally, I'm going to show you a picture here in a second. They're going to get pulled through that chain of proteins. Now, electrons are energy. So as they get pulled through those proteins, they're providing energy for active transport. As the electrons pass through, that's going to power the pumping of hydrogen ions. And so remember, if we're pumping them, it means we're moving them from low to high. And so what we're doing is we're actually storing what we call energy in the form of a concentration gradient. We are pumping these hydrogen ions where they don't want to go. Now, I want you to think about a river for a second. And I want you to think about 2 types of energy. We have potential energy and we have kinetic energy. Now, if you're thinking about a river, there's energy in the flow of the river. And we would call that kinetic energy, the energy of motion, right? If you put a dam up and you block the flow of the river, that is actually going to store a lot of energy. Because what does that water want to do? It wants to flow, okay? So damming the river stores energy. Letting the river flow releases energy. There's going to be stored energy and there's going to be kinetic energy. And this is what we have going on here, okay? Remember, it takes energy to make ATP molecules. So the electrons get moved through the proteins. As they pass through the proteins, it causes hydrogen ions to get pumped. And what we do is we store energy. This is essentially like damming the river. Remember that molecules don't like to be concentrated. They want to be evenly spread out. So when we pump lots of hydrogen ions against their gradient, like we see in the picture here. So here's my electrons. They're flowing through these proteins. All of these proteins have specific names, which you don't have to know. I want you to notice that those electrons are doing that because they're going to oxygen, okay? So the electrons move through the proteins. As they move through the proteins, that powers pumps So you can see I've got hydrogen ions that are getting pumped. And so there's a bunch of them up here. There's not a lot of them down here. So these hydrogen ions are getting pumped to one side of the membrane. And when we do that, it's storing energy. Okay, we're going to come back to this last half here in just a second. So for now, electrons get moved through the proteins toward oxygen. It's that final electron acceptor. As the electrons move, hydrogen ions get pumped, and that is going to store energy. All right, so now we actually have the ATP production, and it comes at the end of the electron transport chain. So at the end of the ETC, there's another protein called ATP synthase. Now notice that ASE, this is an enzyme, okay? Now ATP synthase is going to be the protein that helps us make lots of ATP. This comes from the word synthesis. Literally, ATP synthase is the protein that's going to help us make lots of ATP. So we have pumped lots and lots and lots of hydrogen ions to one side of the membrane. So we dammed the river. We have stored energy. Now, once we undam and we allow the river to flow, there's energy in that flow. And that's what we're going to do here. This right here is my ATP synthase. It's a big chunky protein. And it's going to be a channel that allows hydrogen ions to flow through it. And so as those hydrogen ions flow downhill, there's energy in that flow. And that energy is what allows those phosphates to be added to ADP in order to make lots of ATP, okay? So we're going to get lots of this energy right here as these hydrogen ions flow down through that protein, okay? Now, at the end of the chain, remember that those electrons are going to oxygen. We also have some hydrogens that are going to join up with that. And this is how we get water as a product of cellular respiration. All right, so if we go back to that original equation, we had glucose plus oxygen produced carbon dioxide. We saw where that came out. It also produced oxygen. This is where this comes out. And then we also produced lots of ATP, and we've kind of generated that along the way. All right, so here's the final slide. Okay, what went into the electron transport chain? Well, oxygen gas had to be present. Remember, it was driving the whole process. We had electrons that were coming from those electron carriers. They were getting moved through the ETC, or moved through those proteins. And we needed those hydrogen ions that were also being carried. So as the electrons moved through, hydrogen ions got pumped. And then what did we make? Well, we made lots of ATP, about 32. So you can see that this number is significantly higher than 2 that we got in the other processes. So with the help of oxygen, we generate a lot more ATP. If I started giving you 30-something cents at a time, you would get to that candy bar a lot faster than if I was giving you 2 or 4, okay? So we also produce some water at the end. You can see that the electrons and the hydrogens join up, and water is a product, okay? All right, so here's our final count, our kind of summary. We had 3 main processes. We had glycolysis that happened in the cytoplasm. It gave us 2 ATP. We had the Krebs cycle that happened in the mitochondrial matrix. It also produced 2 ATP. And then we had the electron transport chain, which is in that mitochondrial membrane on the inside, those Christie folds. And here we got about 32 ATP. So from one glucose molecule, this is for one sugar, y'all. Keep in mind how many sugars you must eat when you eat a carb. If you eat a honey bun, or if you eat rice, or if you eat a potato, you've got tons and tons and tons of glucose molecules. So these are just the numbers for one molecule of glucose. We have way more than that in our cells, okay? But for that one molecule of glucose, with the presence of oxygen, we got about 36 ATP. It's not a perfect process. Sometimes we get a little more, sometimes we get a little less, okay? Now, in addition to that, we produced the water. That came at the end of the electron transport chain. And we also exhaled carbon dioxide. That came out in that intermediate step in the Krebs cycle. So if we put all of this together, here is our reaction for cellular respiration. We get glucose in the foods that we eat. We break them down in the presence of oxygen that we breathe in. We exhale carbon dioxide as a waste product. We also produce some water. But most importantly, we get lots of ATP. Now, that was the aerobic pathway. What happens if we don't have oxygen? Okay, if no oxygen is present or available, instead there are what we call anaerobic pathways. All right, so remember that oxygen is necessary for the Krebs and the ETC to happen. If there's no oxygen, those pyruvates will not go into the mitochondria. And so instead, we have an alternative pathway known as fermentation. Okay. Now, fermentation does not make ATP directly. Instead, what it does is it allows glycolysis to continue. So remember, we're in the cytoplasm here. And so glycolysis gave us 2 ATP at a time. Not very efficient, but 2 ATP is better than no ATP. Okay. Now, what your fermentation actually does is it recycles electron carriers so we can keep doing glycolysis. All right, so there's 2 types. We have alcoholic, and this is the process that produces alcoholic beverages, okay, things like wine and beer. And so what happens in this case, so we started off with glucose. In the process of glycolysis, we made 2 pyruvates. And remember, we got these electron carriers. Well, in order to keep doing this right here, we have to have those electron carriers. So what happens is this pyruvate is ultimately going to be turned into ethanol or ethyl alcohol, okay? So ethanol is produced from pyruvates. And in that process, we lose a couple of carbons in the form of carbon dioxide. Now, this right here is why we use yeast to make bread. And you know how your bread dough will puff up and rise? It's because it's releasing that carbon dioxide. So that's what makes that bread dough rise. So what happens is the glucose is broken down into pyruvates. If oxygen's available, these pyruvates would have gone into the mitochondria. But if oxygen is not available, one pathway is that pyruvate could be turned into ethanol. And then we lose some CO2, but we recycle those electron carriers. And so this allows us to keep doing glycolysis and to keep doing glycolysis and keep doing glycolysis. Now, this particular process is not reversible because once those CO2s are lost, those carbons are gone. So this is pretty much a one-way pathway. Now, the other type is called lactic acid fermentation, and this can actually take place in your cells. Bacteria can do this, and this is like what we do to make yogurt. Your muscle cells, when you're exercising and you start to run low on oxygen, you can do lactic acid fermentation. Now, you have to understand that animals cannot survive off of lactic acid fermentation. We can do this in times of need, but if you go for long periods without enough oxygen, you're going to be dead, okay? So we can only use this for short term and only in those muscles when they start to run low. We can't do this for survival. Now, in this case, we still started off with glucose. In glycolysis, they were turned into pyruvates, and pyruvates get converted into lactic acid. Now, this is actually reversible because lactic acid, we didn't lose any carbons. So if oxygen becomes present again, these lactic acids can be turned back into pyruvate, and they can go that aerobic pathway. But for times of need, we can produce lactic acid. And you may have heard that word before. You may have heard of lactic acid buildup in your muscles, and that's what can cause fatigue and soreness. And so that's just one of the ways your body's telling you, hey, you're tired. You need more oxygen, okay? So this happens in some bacteria and animals, whereas the alcoholic, we can't make alcohol in our cells, but yeast can. And that is going to take care of the process of cellular respiration. So once again, I know that's a lot. I know it's very technical. You're not going to get this the first time around. You're going to have to spend some time with it. We're going to do some practice activities. We're maybe going to do some virtual labs, and hopefully you can get a better understanding. But like I mentioned in the PowerPoint, please pay attention to those big ideas. The 3 major processes, what's going in and what's coming out, and hopefully you'll be able to do okay with that. We'll be doing some practices over the next several days. If you have any major questions or concerns, be sure to get help, and hopefully I will see you soon. Y'all have a good day.